Next session. So here I would like to introduce Dr. Muthukumaraswamy Karthikeyan. He is a senior principal scientist in the Chemical Engineering and Process Development Division of CSIR National Chemical Laboratory, Pune. And he holds PhD in organic chemistry and working in the area of chemoinformatics, its application in drug discovery and design of novel material for the past 30 years. He is also the recipient of Boyscast Award by Department of Science and Technology and Overseas Associate Award by Department of Biotechnology. He is selected for 2015 Open Innovation Challenge Award by Thomson Reuters and Stanford University. He has authored a unique book, textbook on practical chemoinformatics, Springer 2014, and published about 30 research papers and holds several national and international patents in the area of chemoinformatics for drug discovery and material design. Currently, he is working in the area of harvesting large-scale molecular data associated with medicinal plants and their bioactivity, artificial intelligence application for cancer cure, and precision agriculture applying artificial intelligence and robotics methods. He is associated with NIPER Kolkata as an adjunct faculty in the area of chemoinformatics education and research. I welcome him to deliver a talk on artificial intelligence in cancer using open source toolkits. I welcome you, sir. Over to you.
I am unable to share my screen.
Yes, sir, just a minute, sir.
I hope you are able to see the screen.
Yes, sir.
Okay. First of all, I'd like to thank the organizers for inviting me to give a talk on artificial intelligence in cancer using open source tools. Okay, yeah. So I stopped the camera because of the bandwidth issues. I think if you have any problem or if you have any questions, you can feel free to ask me. I will be presenting some of the technical aspects of AI in cancer based on the work which I have done last 3 years under some sponsored program. We are working on practical aspects of machine learning, deep learning, etc., and how to apply these machine learning, deep learning techniques for drug discovery. And I will be focusing on specifically how to apply these techniques for cancer research. So as Professor Sastry sir said, it is very clear that all the medicines won't work for all. So cancer specifically is person dependent and gene dependent and heredity dependent. There are so many factors responsible for uniqueness of this particular disease. Today, what I will be covering is what are the traditional versus modern drug design, and what is the general approach for drug discovery versus personalized medicine, and then drug discovery pipeline, which Dr. Sastry already mentioned, and I will just a little bit touch upon it. We are working in the chemoinformatics for last 30 years. I will be discussing the open source tools, which is very much used in our research for integrating all the open source tools to build a software. We call that is a CCCTK, that is Compute Cure Cancer Toolkit. I will be showing some screenshots of the work we have done, and I will be touching upon the big data, AI, HPC, text mining, data mining. And then for any research, the scientific data is very important, especially the open data. So we need to get the cancer specific open data, how to use those data for learning purpose using the machine learning and deep learning techniques. And then for the user friendly software we have to develop so that it can be a chemist, biologist, medical practitioner, anyone can use this. And then I will be talking about what is the role of AI and what the future holds. This is the agenda around which I will be presenting. Okay, so okay, I'm sorry, I'm not able to see the screen. Okay, fine. Therapeutic targeting of hallmark of cancer is shown. Cancer is a very complex disease. Okay, so now I will not go into the biology and mechanistic aspects of the treatment of cancer. There are several textbooks and information available. I will be focusing on how to design anti-cancer agents, and then how we can use AI for that purpose. So inhibition of tumor growth by targeting downstream signaling elements is shown here. So it's a very complex procedure. Now, there are several types of inhibitors can be developed, as is shown here, JAK inhibitors, SRC kinase inhibitors. There are several types of inhibitors, and then there are several pathways involved for cancer disease. And then we have to stop the cancer cells, specifically growth of cancer cells specifically, without affecting the normal cells. Okay, so then treating cancer is very, very complex, and it involves several targets. Especially whenever you take a drug in your body, there are about 5,000 targets are there. So how to specifically send the drug to bind or inhibit or interact with particular target without affecting others is a very big challenge. Okay, so then AI comes into picture. So Dr. Biswas told that treatment of cancer is very, very complex. People use surgery, people use radiation, people use chemotherapy, then anti-neoplastic agents which should distinguish normal versus cancerous cells. So these are all the complexity involved in treatment of cancer. Today, cancer is spreading like forest fire in the world, and we need to somehow focus on developing treatment or drugs to solve this problem. There are several common drug toxicity associated with this cancer treatment. So why I am giving this known information? Because once we know the problem, as Dr. Sastry said, we can find the solution. Okay, until unless the problem is very well defined, we can't do anything else. Okay, so now these drug toxicities are very important to understand. Okay, so then there are several types of anti-cancer agents. For example, alkylated drugs, anti-metabolic drugs, drug derived from plants, hormone antagonist, antibiotic drugs. I will quickly go through the type of drugs or treatment used in cancer. Then we will go into details of how to design the better ones. Okay, and then currently there are these are the anti-cancer drugs which is very frequently used today for several types of cancers, right? Okay. So now when you take a cancer drug, we need to know the chemical nature, mechanism of action, resistance, cell cycle specificity, toxicity, and then therapeutic use, and then any other issues associated with the drug. So every drug should go through this process. Then basically by going through this process, we are collecting the information that become input for design of future drugs. And especially drug discovery in the age of AI, especially personalized drug design, one key won't fit for all the logs. So we have to find specific key for specific target with respect to specific mechanism of the specific disease. So in this context, learning is very important and automated learning is very important. Today, with the help of technology, we will be able to do it. So nowadays, everybody is talking about artificial intelligence and machine learning, how it helps to build the computational models which learns from the experiments or information, and then how to design a novel drugs based on target specific drugs. That is what everybody is today attempting to do it. Here I am showing for the benefit of people. For example, money as a cash, anybody can use it. But when the money is available in your personalized cards or UPI payment, only you can use it because you need authentication. Exactly, in the context of cancer research, all drugs won't fit for the diseases. So there are several cancer drugs, but it may not work for all the people because of the diversity within people, genome difference in the people. So we need to collect those informations. What is the variation in the people? For that, we need to give a specific drug so that you'll be able to give the specific drug which cures specifically without much side effects or other issues. This slide I have to show because whomever talks about drug discovery and development discuss about this. So from identifying disease, filing means discovering a new drug, okay, and then it goes through several testing purpose and finally it becomes a drug. So where the cheminformatics plays an important role, okay. So cheminformatics plays an important role in combinatorial chemistry, molecular modeling, virtual screening. So that I will be focusing only on the molecular informatics aspects of drug discovery. Then can we use AI for that purpose? So slightly to touch upon what cheminformatics for the newcomers, everybody knows about bioinformatics, but in cheminformatics now it is emerging area. So cheminformatics helps to collect the informations in large quantities, especially chemical informations. Using cheminformatics, handling molecular structure is very easy. Whereas in bioinformatics, you can use sequences, textual informations, etc. But whereas in cheminformatics, the molecules are in pictorial representations. Understanding the chemical structures, transforming into a reusable format is a very challenging problem. Today, we have 50 plus years of data. And then over a period of time, we developed several tools and methods. Now, there are several type of people working on this cheminformatics, starting from chemists, biologists, teachers. Anybody can complement this domain. And there are about 35 million chemistry-related publications, which is indexed by PubMed and Chemical Abstract Service, which become a input data for learning purpose. What the machine, if you are telling the computer to learn, from where it can learn? For a child, learning is easy. Whereas for a computer to learn, it needs to have a proper data in proper file format. So these 35 million publications or experiments, which is already available, can become an input for computer AI learning. There are about 15 million patents with respect to chemical data is also available for this machine learning. Now, extracting the chemical and biological relevant information, especially with respect to cancer, and then putting in a proper format so that machine can learn from that. As I'm going to show you how to do that, okay? And then we can use this knowledge discovery for new chemical entity design. And then there are some people who ask the questions in the morning, what are they, how to predict these side effects? Okay, so those things can be, how can we identify the adverse effect based on fragment-based drug discovery? So this, once you make the machine to learn the molecule and bioactivity or the adverse effect relationship, it is possible or machine can give you alert how to design the molecule with minimum side effects or adverse effects. So basically what happens in cheminformatics? Cheminformatics, we are mixing the informations, we are transforming the data into informations and information into knowledge. That knowledge actually helped the AI so that you can design a better molecules or you can make a better decision faster. And then in cheminformatics, it involves several components: design, creation, organization, management, retrieval, analysis, dissemination, visualization, and most importantly, use of chemical informations. And cheminformatics is beyond informatics. So what cheminformatics does is to help to discover the hidden patterns responsible for, in our case, desired bioactivities with respect to cancer, and then physical chemical properties, etc. Right? So now in research, what we do? In research, we try to discover what is unknown. Then only you can publish papers, you can take patents, and then you can protect intellectually. So here we are trying to identify the AI methods to collect the information, analyze the information, understand the patterns, and identify what is unknown. Okay? I will be discussing about that in short while. So using this AI, we will be able to do in-silico experiments, and then in in-silico experiments, we can that AI can guide us which compound to screen, what are the libraries to synthesize, which compounds to purchase from commercial sources. Okay, so because the chemical structures are represented in a pictorial format, searching them from commercial sources is a challenging task. So people use several techniques like substructure search, exact structure search, similar structure search. So I will be talking about the structural similarity with respect to QSAR. Okay. So what we do compound registration databases, and then we do primary and secondary literature screening, and then we do QSAR, and then we also do physical chemical property calculations using these methods, chemoinformatics methods. Okay. Now, in how the chemoinformatics pipeline for drug design. First, you have to identify the new lead structures and optimize the structures, establish the QSAR, and then compare the libraries, definition and analysis of structural diversity. This is also very important because similar molecule will have similar bioactivity. So then planning a chemical synthesis also very important. And then analysis of high throughput data, docking ligand into receptor with respect to the disease of interest, and then de novo design of ligands. So far chemistry has produced enormous amount of data and it is rapidly increasing. There are about 35,000 journals which publishes chemical and chemical informations, which may also include so much bioactivity informations. Because when a person do either clinical experiments or synthesis or bioactivity screening, he usually publishes. AI can learn from those publications. So basically, you are learning from the experiments. Okay, and then you are able to design the new molecules, etc. Right? So the chemical space is huge. It is several millions of molecules. Dr. Sastry also mentioned 110 million compounds already in his repository. So how to search those information out of 110 million? Which one are cancer specific? And then among the cancer specific, which one is there are several types of cancers. Even if it's a top 10 cancers, how these molecules will respond to those different types of cancer. So there AI can be used. That is what we are going to show. Okay. Then actually in computational modeling helps to avoid animal experiments. And then okay, I think there is one problem. Okay. Most importantly, chemoinformatics helps virtual screening experiments to score and rank the molecules and filter the set of compounds. And then he also mentioned about ADMET properties should be predicted. So now due to animal rights, animal cruelty should not be done. So computers are used to predict the animal toxicity models from learning. Okay. So now, okay. So now I'm going to give little bit brief outline about what is a chemical structure. This is very important. What is a chemical structure? How it is represented? What informations can be derived from the way in which you represent the chemical structures? And then how to use this chemical structure representation for predicting the physical properties, chemical properties, and biological properties. And then you can also, once you represent the chemical structure properly, you will be able to plan how to synthesize this compound, what are the known methods of synthesis of these compounds, and then what type of reactions it can undergo. Understanding the chemical functional groups. When it is a drug, what are the possibility it can go through enzymatic reactions so that it, the drug may not be toxic, but when it goes through the biological systems, it gets modified So what are the reactive functional groups present in the systems which makes the drug toxic? So those type of informations can be derived. So representing chemical structure properly is very important. So I will be talking about how to handle the chemical structure properly. Once you know how to handle chemical structure properly, once the computer understands the chemical structures, atom bonds, connectivity, etc., then charges, etc., then it can predict the bioactivity spectrum with respect to known information. So the molecules are represented in literature in several formats, textual format. Even if it is represented as a chemical name, there are several chemical names, synonyms, and there are several synonyms, and then corporate names, CAS registry numbers, many things are there. So now the chemical knowledge, which is from different sources like documents, reports, etc., needs to be understood by the computers. Here, when we call computers, that is a AI system which should understand the chemical knowledge properly, which is coming from different sources in different formats. Then only it will have a ability to understand, build the model, and predict properly. So in this particular case, molecules are represented in pictorial format, in which can have some intrinsic properties. It has a chemical properties and it has a biological properties. When a system is harvesting information with respect to chemistry, one, you can get the bioactivity properties like activity, toxicity, biotransformation, pharmacokinetics with respect to the particular molecule from the published literature. But given a chemical structure, you can also identify or calculate some of the intrinsic properties as the description of the molecule, which will be helping to build the or predict the bioproperties. So intrinsic properties can be calculated, and then with the help of these calculated properties, you are trying to predict the chemical properties and biological properties. This comes under QSIR, QSPR, and QSTR. Okay. Then chemical structures cannot be directly used for calculating the properties. So the chemical structure needs to be understood in the form of structural descriptors, and then machine can correlate the relation between chemical structure and descriptor. And then once it understood from the chemical data, the information, the knowledge, then it is called inductive learning. That means given bioactivity, given experimental measurement, with particular context of chemical structure, the knowledge is built, which is a mathematical model. Now the AI systems can use this knowledge for predicting the bioactivity data for the unknown compounds, because the unknown compound may not have bioactivity data, but from the unknown compound you can calculate the intrinsic properties. The intrinsic properties in turn are molecular descriptors. Those descriptors are applied in mathematical format to predict the bioactivity. I will go into the details in shortly. Okay, so basically you have a chemical structure, and then you cannot predict the properties as shown in this picture. But then you have to represent the chemical structure in 2-dimensional and 3-dimensional, and then you generate the molecular descriptors. There your open source tools comes into picture. There are some commercial software which can calculate the molecular descriptors. There are several open source tools which Dr. Sastry also mentioned, Padel descriptors, etc. So those things can be used. Given a molecular structure, first step is to calculate the molecular descriptors. What is the molecular descriptor? Given a molecule, you are calculating the molecular weight. How you calculate the molecular weight? You calculate based on the composition of atoms in a molecule. So that is the description of a molecule. Once you understand the calculation of molecular descriptors, that is describing a molecule, how many rings present, whether carbon containing compound, oxygen, nitrogen, etc. So those calculations, and then whether aromatic or aliphatic, those informations comes under molecular descriptors. So once you describe the molecule properly, which computer can understand, then with the help of correlation, machine learning, deep learning, you can build the mathematical model. With that mathematical model, you will be able to correlate the property with the molecular structure. Once you successfully correlated, then you can apply those models for the molecules which is not yet synthesized to predict the properties. In our case, we will be identifying anti-cancer agents from the known, by learning from the known anti-cancer agents. Then we are trying to predict the novel anti-cancer agents with specificity, selectivity, etc. Right? Now, in open source context, there are CDK descriptors and Pathal descriptors are freely available. Given a molecular structures, you will be able to calculate molecular descriptors, which is very important because computer can correlate number with number. So your bioactivity is in numbers, micromolar, nanomolar, in that particular format. Okay, then your molecule also should be represented in numeric format. So mathematically, you are correlating number with number. Then that is where your machine learning, deep learning comes into picture. Okay, so that AI system is nothing but correlating a description of a molecule with bioactivity or property or toxicity of interest. So how to describe the molecule? Molecule can be described using descriptors. What are the types of descriptor? Your constitutional descriptors, topological shape-based descriptors, physicochemical descriptors, quantum chemical descriptors, electrostatic descriptors, and drug-like indices, which the previous speaker spoke about, rule of 5, rule of 3, etc. So there are so many types in which you can describe the molecule. Then there are several algorithms like genetic algorithms, neural network, principal component regression. There are several statistical methods and algorithms which can correlate the description of the molecule, which is descriptor, with bioactivity of interest. That is where your AI plays an important role. Now, the first step is, as I mentioned, where the data is coming from. The data is coming from literature. Can you read all the publications with respect to cancer? There are about 2 lakh research publications with respect to, say for example, one type of cancer, say breast cancer. So if you go to PubMed, you search cancer, then you will get millions of hits. Are you going to manually read the papers word by word, character by character to extract the chemical informations and bioactivity data? There Chemoinformatics and AI systems come into picture. So as Dr. Shastri mentioned, there is a text mining tools are available, which comes under NLP, natural language processing. What the NLP does? Next, text mining is very, very important in transforming your molecular data and bioactivity data in a mathematical format. So text mining is a method where the computer understands the abstract or full text in word by word, as a chemical, as a protein, as a disease, as a drug, as a bioactivity, etc. So using the named entity recognition, the text which is present in the abstract or the publication is converted into or recognized as chemical, protein, disease, and bioactivity of interest. Once these patterns are detected, then the systems understands the correlation between your molecular information and bioactivity information. There are several parts involved in text mining. So basically in text mining, in our case, since we are working on personalized medicine with respect to cancer, we need to identify the protein, gene, drugs, and disease, especially type of cancer associated with this particular drug from the literature. So the text mining part, that word by word recognition will lead to building the repository of or a resource of this protein, gene, drug, disease network. It will form a network. It will make a bio network. Okay. So people talk about polypharmacology, and here we are building the biological network where molecule play an important role. Once you have the molecule and the protein association with respect to disease of interest, in this particular case we are talking about this cancer, so then we can try to find the frequency in which the similar information is present. So now let us go into the detail. Once you got the chemical structure, when you look at a pictorial format, it may have from the literature you will be getting biological properties, chemical properties, physical properties, and electronical properties. Now, when we are talking about anti-cancer agents, we know that the compound is anti-cancer, that is biological effect, then which should be a function of molecular structure. Then here I'm showing some simple example for the beginners. There is a chemical structure called paracetamol, which relieves the pain, which everyone knows. There is a structure called cyanide, which is, we know that it is toxic. There is another structure, saccharin, which is having sweet taste. So basically, if you look at all the molecules, their atoms and bonds, why they are different properties, different toxicity. That's what computer should understand. How you can, with given this molecule and property, with the help of descriptor, the molecule will be able to understand what is the constitution of this particular functional groups or fragments in the molecule responsible for this particular bioactivity. Then you can be able to build the mathematical equation. The biological effect is a function of chemical structure. Basically, a property X, which can be a toxicity, property, activity, anything. It can be a function of chemical structure. So this part is very, very important for the computer to understand. This is where AI comes into picture. Once the computer understands the property is a function of structure, because computer is nothing but a mathematical big calculator or something which handles the mathematics very fast. Today, the high performance computing with petaflops computing and terabytes of storage available, so we can be able to handle lot of information where property X and function of structure is fed into the systems. I am going to details about how the 3 million experiments of this activity of anti-cancer activity with respect to function of chemical structure tested on this anti-cancer activity was given into the computer, and how it was used for building the mathematical models. I will go into the details. But once you understand this, property X is a function of chemical structure, then once you can make sure that the computer understands this, you can build the predictive models. Okay, so then basically, as I said earlier, molecular structures can be described in mathematical or numeric form that can lead to molecular property predictions. So then people say that, okay, one model cannot fit for all. So for every type of activity or bioactivity, in this particular case, oral bioavailability, we have to build a separate equation that is a function of different set of descriptors or molecules. Then similarly, cell permeability. So we will take a compounds in different region and then use those set of compounds for machine learning. Then when people talk about anti-cancer and toxicity, we need to have a set of compounds which are toxic to normal cell versus cancer cells. So we need to have a separate set of molecules. Okay, so this is how you can have a set of molecules and set of descriptors which describe that particular property. Then you will build several mathematical equations. Each equations will help you to predict that particular property of interest using this machine learning and deep learning techniques. So now I can say that molecule saccharin can be represented in 1-dimensional, 2-dimensional, 3-dimensional. You can derive different type of information the way in which you represent the molecules. Once the molecule is extracted from the literature, say for example, it is extracted as a 1-dimensional molecular compositions, you can still calculate number of atoms present and molecular weight and etc. etc. When it is represented in 2-dimensional, it can you can derive these informations how the molecule is described, whether it is a ring containing compound, etc. So now when your molecule is represented 3-dimensional, this is different type of information is again collected. So all these combined information helps to build the mathematical model which can predict the anti-cancer activity. So once you understand the relation between molecule and the bioactivity, now you can design a molecule. This is a manual method. So similarly, as we understand what is the intelligence, Dr. Sastry told. So intelligence is learning. Then we are learning from the information which we get. Then once we learned, we can do some activities, apply that learning for designing something. So similarly, what happens in this learning? What is the learning curve? So you have a set of compounds, say anti-cancer compounds, and then for every anti-cancer compound, you describe the molecule in mathematical or numeric format. Then you build the QSIR model. Okay, you build the QSIR model, and then you validate the initial model. This is the QSIR pipeline, where first you find the list of compounds and the anti-cancer activity, and then you calculate the molecular descriptors using your open source tools. Then you build the QSIR model. So how you validate the initial model? So your expected value or experimental value should be matching with the predicted value. So if it is matching, very good, your model is very good. Still, you can optimize the mathematical model by adding and removing descriptors. You can refine the mathematical expressions, and then you can get the better bioactivity. Say, for example, your anti-cancer agent IC50, LC50, GA50, growth inhibition 50. For example, GA50 values you are measuring. So your experimental value matching with your predicted value, you can say that your mathematical model is perfect. Okay, till that stage you have to keep learning. But unfortunately, because of the molecular diversity and the experimental complexity and how the molecule respond differently, it is difficult for getting the perfect model where you are able to predict exactly the experimental value. Whenever the experimental value is exactly predicted by your system, then you should doubt your system. Okay, so because in biology it is not like that. So we have to learn, we have to understand what went wrong. Okay, so there are several this is the first I've shown you how you learn from the molecule and the molecular descriptors, and then you build the mathematical model, and then you are able to predict. What are the methods used? I told earlier that genetic algorithm is one method. So there are several machine learning methods, data mining methods, statistical methods, pattern recognition methods. These all the methods are inbuilt into the system. So when people earlier they called it is a machine learning. After that, now it is become deep learning. When all this thing learning comes, then you will be, that comes under part of your AI system. Okay, so now what we did in NCL, what is something special which we did in Chemoinformatics component? What is our contribution in this Chemoinformatics for designing a new molecule from the known molecule? So how you can design a new molecule from the known molecule? So what we did was, so identify the molecular scaffold. So here I'm showing an example. This input molecule is published in the literature. Then that computer softwares which we developed or algorithms which we developed, able to recognize the molecular scaffold, ring containing compounds, and then it can be classified into three types: basic scaffold, extended scaffold, and framework. Why? Because from this scaffold you will be able to build the virtual library of novel molecules. Okay, so that is what. Okay, so now if you look into the studies, we took around 70 million compounds from PubChem, and we extracted using this our technique, Chem Screener. So we were able to identify 800,000 scaffolds from this PubChem, which is not necessarily all active compounds. And then there is one more database called Campbell. From that Campbell, we identified around 54,000 scaffolds. So now, instead of looking into 800,000 scaffold, which are diverse compounds which may not have biological significance or relevance, now we have to focus on scaffolds which has a biological significance. Among the 54,000 bioactive scaffolds, we have to find out what is the anti-cancer specific scaffolds. Once we understand the list of anti-cancer compounds and anti-cancer specific scaffolds, we will be able to generate that virtual library of anti-cancer like compounds. That is very important. So I will be showing how we actually did this work. Okay, so when you do the molecular analysis, you can find the molecular diversity. That means similar compounds in this chemical space come together. We can find that there are some white space where you can identify if a molecule comes into a non-crowded area, then you can say that that compound is novel. So this can be used to identify the novel molecules from a collection of diverse compound. Here we put all the some of the bioactive class of compounds, and then we try to find out what is the scaffold responsible for what type of bioactivity. So once we try to find the scaffold diversity with respect to specificity of the disease of interest. Okay, so then we took anti-anginal, anti-diabetic. So there are scaffold specificity with respect to these therapeutic categories. So once you have that anti-anginal, there are about 40-50 scaffolds responsible for this anti-cancer. We don't need to worry about other scaffolds. We can focus on those anti-cancer scaffolds. So what happens after that? The next step is, once you identified the scaffolds, then we have some modules called Scaffoldopedia, which goes through a pipeline of activities where the scaffold and functional group identified from anti-cancer agents are reconstructed to build the virtual library of compounds. So once the virtual library is generated, then it goes through TPC score. What is TPC score? T refers to toxicophores, P refers to pharmacophores, and C refers to chemophores. What is the toxicophores, pharmacophores, and chemophores present in your molecule? And then what is their role in your virtual library of the compound? Once you identified the risk associated with your virtual compound, then you can use some screening protocols to remove some of the toxic compounds. The workflow is like this. First, you understand the bioactive materials, for example, in this case, anti-cancer agents, and then these molecules go through several steps in a pipeline. For example, building the virtual library, and then calculating the molecular descriptors, and also calculating the molecular fragments in the context of pharmacophores, toxicophores, and chemophores. So based on that TPC scores, now you will be able to find out whether the compound is active or inactive, toxic or non-toxic. Then it goes through second level of QSIR studies, where you can able to build the mathematical model and predict the anti-cancer activity with respect to specific cancer type. Say, for example, breast cancer, colon cancer, etc. So we need to build the mathematical model specifically for type of cancers. Okay, so what is chemophores? So if you take a collection of compounds, say 50 compounds, what are the fragments which is present, especially chemical fragment present in all the compounds with respect to the particular class of compound? So in this particular case, what are the chemophores present? So then this become a model. So if you want to design a new molecule, these features must present because these are the anti-cancer specific fragments. So this is where the machine learns, and then it helps to screen or design or shortlist the molecules based on these informations. Okay, so this is not static informations. Today I have 30 compounds with respect to particular type of cancer. Maybe in 5 years from now we may have so many other compounds. So then this pattern will change. The functional group or fragments collection and then statistical significance of that particular functional group present in a collection of molecule will change. Accordingly, the mathematical model will change. Then the prediction also will change. Okay, so using this type of information, so now you will be able to shortlist what is the active compound, inactive compound, what is the toxic compound, etc. using this SOM type of analysis. Okay. Then you can also find the molecular similarity with respect to molecular diversity. Similar compounds comes in one space, and diverse compounds you can find in this space. Then we also used NMR spectroscopic data of the drugs. So we took around 1,400 drugs, and then you try to make the simulated NMR. This is H1 NMR of simulated NMR of all these cumulative NMR of 1,200 FDA drugs. Now we know that this can be used to find one, like Lipinski rule of 5, whether drug-like nature. How we can use it? If the peak present, if the simulated spectra contain the particular peak in the region where this FDA drugs has these peaks, then we can say that there is some similarity with respect to drug likeliness. So similarly, we did for this C13 spectra simulations. For given a structure, we were able to generate the C13 spectra. Actually, you can also experimentally measure these spectra H1 NMR and 13C NMR. Now, this model can be used looking at the spectra itself, whether the anti-cancer like features are present in your spectra, because spectra is nothing but how the atoms and bonds positioned in a molecule. Okay, so since QSIR is also talking about similarity, here by spectral similarity we will be able to tell whether this compound is anti-cancer like or not. This will be very, very useful when you extract a molecule from a natural product, because a natural product, now many natural products are used as anti-cancer agents. So people mentioned about taxols, palitaxols, etc. So there are several natural products which is extracted from plant sources. Now looking at the spectra from the experimental spectra characterization of this particular compound, we can use the spectra itself as an input for identifying whether it has some anti-cancer properties or not. Okay, so we did some studies on this. We published some of the papers. Basically, once you have the molecule which is having anti-cancer activity, now what we can do? Now we can use this one to dock against the anti-cancer compounds against the cancer-specific targets. Okay, some say for example kinases. So we can do the docking and find the second level of screening whether the compound is one it is bioactivity. What is the GI50? Growth inhibition factor, what is the IC50, what is the LC50, what is the TC50. These are the QSIR based studies. Second step is whether the compound is anti-cancer, whether it is toxic, using our TPC scores, whether it can be eliminated in a very rapid manner, because TPC score actually tells in binary format. Computers are very good in handling binary information. So the binary fingerprint generated for the molecule for the toxicophores, chemophores, and pharmacophores are screened rapidly so that the molecule again pass through the filter. First step is whether it is anti-cancer or not. Second step is how much good it is. Third one, whether it is toxic to the normal cells or not. Then it goes to whether which is having any target specificity, selectivity, and then whether it binds. Because while doing this protocol itself, we will come to know whether the compound is soluble, whether it is other characteristics required to be a drug. So then it goes through 3 levels of screening. Step 1, drug likeliness, lead likeliness. Step 2, anti-cancer properties screening using QSIR modeling. Third one is docking studies. Okay, so now I will spend some time with respect to the software which we built over a period of last 2 to 3 years, which is funded by CSIR. This is the Compute Cure Cancer Toolkit. Okay, so what it contains? This tool is basically mainly based on, we are using machine learning, deep learning, and AI techniques. Okay, so the first step is data. The data is coming using text mining and also from extraction of information from commercial and open databases. Okay, so we use NIH data, National Institute of Health, which provides a huge data with respect to cancer, National Cancer Institute, NIH, and then PubChem and PubMed. These are the sources from where we started our research, because we can't do the experiments blindly. So first we need to learn. The AI gives an opportunity to learn from what is already known. Then in that process, to train the system, first we took all the data available with respect to cancer from different sources: literature, molecular data, bioactivity, and then cell level screening, different type of cell lines. Say for example, 800 type of cell lines are there, and then 800 cell lines, 800 type of cell lines. And then we have to find the 60 specific cell lines with respect to top 10 cancers. So that is what our first mission was. Our first mission is to find out the molecular bioactivity screening context, molecular and anti-cancer activity of the compounds of 3 million experiments. Okay, what is 3 million? There may be compounds common in so many cases, and these cell lines may be common. So now we have to find the cell line specificity with respect to the molecular diversity. Okay, so that is our first step. So this information is fed into the systems. Now what we did, we put data mining tools, big data analytics platform, machine learning, deep learning, and we also used Bioinformatics. All the tools, whatever is open source tools available which can handle molecules, we integrated into a open source platform. This is basically the architecture of the tool. Okay, so purely open source, but unfortunately it is not yet published. It is supposed to be completed by March, and due to this pandemic it is getting delayed. So we are looking for the extension of this project so that by next year or something it will be released to the common man. So it contains chemoinformatics tools, bioinformatics tools, text mining tools which can handle literature mining, patent mining, and then which can handle sequences, structures from PDB, and then it also has structure activity relationship, and molecular mining, molecular dynamics is going to be integrated using NAMD, NAMD, this GROMACS type of open free softwares. And then molecular docking we tested with Vina software, and then other commercial softwares also we are trying to link with proper licensing. And then we have inbuilt machine learning techniques to build the QSIR, QSPR, and QSTR models. Then basically it is a combined molecular informatics platform for predicting whether one, drug likeliness, cancer, anti-cancer agent specificity, selectivity, etc. And then what is the target specificity of the particular compound. So what happened here? So we put 3 million experiments into the system. Then we also took the clinical drugs with respect to anti-cancer agents. Now you put the clinical drugs, then you extracted the molecular scaffold with respect to the anti-cancer agents. Now then you, as it is shown here, we built the network of scaffolds and functional groups, and then that forms the virtual library of novel molecules. Once you have a virtual library of novel molecules, then we use inbuilt open source based descriptor calculating tools, say for example Padel, and there are some RT-Kit. Many things are there. So this software is developed using Java platform, and even though it is in Java platform, it is capable of connecting with Python, C++, any executable files, command line utilities. So this software is capable of handling connecting the softwares, other sources, other databases. It is integrated with MySQL, PostgreSQL to handle large data. It also has a connectivity with NoSQL systems. Okay, so it can handle XML, it can handle CSV, it can handle textual information, even PDF files. So it extracts the information, it identify the molecules and bioactivity. Then from the molecules, it can extract the scaffolds and functional groups and build the virtual library. Once the virtual library is built, then it can undergo the pipeline of calculating molecular descriptors. And then once the molecular descriptors are calculated, which is very critical, then it goes to the machine learning, deep learning tools to build the mathematical models. We used about 7 machine learning methods and 3 deep learning methods. And then for this type of calculations, we need high performance computing. We used GPU enabled systems and high performance and high storage base. We recently established some 100 terabytes storage facility where this information is being processed, along with very high performance GPU enabled systems, so that you are able to handle millions of calculations in shortest possible times. We have a cluster of computers which can, there are about 24 servers continuously working on this text mining, data mining, and machine learning techniques. Then you can use the mathematical model to predict the anti-cancer activity. So basically, the input is coming from literature or known databases like NIH or NCI. Then it also comes from drug banks where FDA approved drugs, then Campbell, etc. Now you identified anti-cancer agents. From anti-cancer agents, you identified the molecular scaffold specific to this particular cancer type. Then it goes through virtual library and building mathematical model, and then you are able to tell how to design a new molecule from the known molecule. That process is complete here. What is the next step? So this is the overall view of the anti-cancer agents, whether it is synthetic, natural, commercial, whatever the sources. So it is also a system can extract information as and when it is available. It is not a static system. It goes through dynamic processing. Then it goes through these calculations automatically, and then goes through the pipeline. And then finally you end up with a molecule which is anti-cancer, which is specific and selective. These are the recently with respect to, we applied the same technology for our anti-COVID research. Okay, because the pipeline is already available, I just want to show you that there are about 50,000 compounds with respect to anti-cancer, antiviral agents given by CAS, which is first time in the history of CAS. They released 50,000 compounds. Then we processed them. in the context of anti-cancer. Since the pipeline was ready for anti-cancer, it was very easy for us to use it for antiviral activities. So this work is going in parallel based on the pipeline we already defined in software. So basically, the system is able to identify or characterize the class of compounds and what are the molecular descriptors. This is not a one molecule; it is a set of molecules. So for a set of molecules, say for example antiviral or anti-cancer, so what is the range in which the descriptor should come? Say for example, Lipinski rule of 5 came based on some particular range. Say molecular weight should be below 500. They mentioned about hydrogen bond donor, etc. Here we are talking about 180 molecular descriptors. So in that 180 molecular descriptor space for these anti-cancer agents, what range should be there? This range become a model. So for a given compound, whether the range is there or not, particular range is there or not. So based on that range, we will be able to tell whether the compound is anti-cancer, and if it is anti-cancer, for which cancer it is likely to be better. Okay, so this the tool as integrated with Chemoinformatics tools. So because we are partnering with Chemoinformatics software company, usually they gave the software in a academic price, and now previously they used to give free. Now it's nominal price they are giving. So we integrated that drawing tools because we don't want to reinvent the wheel. So it's very easy. It also has so many molecular descriptor calculating tools. So we use this Chemoinformatics tool as the front tool. All other information is in the back end as a open source tools. Okay, it's a mixture of academic software with open source tools. Okay, so you can also use equivalent other drawing tools which is freely available, but we found this is very useful, and the academic licensing is very compatible for our work. Okay, so you have a drawing tools, Chemoinformatics component, you have Bioinformatics component. Now we are actually integrating the TCGA, the Cancer Genome Atlas. So TCGA data contains about 400 terabyte of information with respect to cancer. So now what it contains? It contains, so what is the effect of a molecule in a patient? Okay, so then you can, that information also we are capturing at the end of the day. If the patient, what is the type of patient, whether it's a young, old, male, female, what type of cancer, what is the level of cancer, cancer level 1, 2, 3, different stages of cancers are there, in what condition, what type of medicine given, what is the concentration, then what is the final outcome. Okay, so what is the final outcome, whether the patient is alive or dead, and how much time the medicine is administered, and then what is the scenario. So of course, except this patient name and other private information, we try to capture the information with respect to building models, so that the next drug which should be effective based on the what is learned from clinical data. So that clinical data is also integrated in these systems. So molecular data, text mining of the literature, and then clinical data. At the end of the day, Dr. Biswas said how it is going to help the clinicians. Can we develop a AI system to help the clinicians? So if the system which you are building is not helping the clinicians at the end level, it is no use. So we are trying to link the research, discovery, and clinical data together, so that this system is also can be used by clinicians with the background of what literature or scientific evidence responsible for making that drug information, etc. Everything is integrated. Okay. Now this I am trying to show you that this is the way different type of cancer data is coming. So breast cancer, CNS cancer, colon cancer, all data is now captured, and then how the IC50, GI50 varies, assay data, bioassay data, everything is integrated into this system. It has a input data, it has a input molecular input system. It also has a data extraction system automatically from the published or open domain data. Okay, so this NCI data is directly input into the systems. Then the NCI data, National Cancer Institute data, contains this bioassay data with respect to type of cell lines, and then actual chemical structure. So now the chemical structure data is there and bioactivity data is there for 3 million experiments. So this for 10 types of cancer. So now this AI system is able to understand what is the variation in molecular structure responsible for variation in your bioactivity. This one slide shows actually in the system what is the variation in structure and what is the variation in bioactivity. Okay, so now the next molecule when you design, you can easily use this information based on your molecular fragment designs. Okay. Okay, then it also has we built with the bioinformatics component where we can do the sequence alignment. We can download the targets with respect to the cancers. Here the PDBs of all the cancer related proteins are integrated, so that if you want to do docking studies with respect to particular target, you can do dynamically in a high performance computing using this interface. So you select the target of interest, or you type what type of target you want with respect to and what type of disease related, especially suppose breast cancer related target only you want to do docking studies and want to see the performance of the binding energy with respect to the class of compound, you can do that. Now you can shortlist the better compounds from these molecular studies and also molecular docking related studies. We built with several machine learning, deep learning techniques. Here we used Vika software, which has so many machine learning tools, and we also used TensorFlow, etc., which uses the deep learning systems, and then in the GPU systems. So then different type of statistical and machine learning, deep learning techniques are integrated. So the data goes input, and using different methods, what is the models built? Which model is very efficient? The best model is the one which is able to reproduce experimental data is the one. So we have to shortlist the models which statistical techniques or machine learning or deep learning techniques which able to reproduce the experimental results properly. Okay, so that is very important. So taking these 3 million experiments, giving to different statistical techniques, coming up with prioritizing the statistical tools which gives the better results for 10 type of cancer. This is what the entire tool is all about. Now, what else we can do? So now, for a chemist, so they make a list of compounds. They may go through all the published literature with respect to the research or not. Now, say for example, they have some 10, 15 compounds in their collection in their laboratory. So they synthesized thinking that it can be antiviral or antibacterial, antifungal. So now that compound, you want to know whether it's anti-cancer. Because when Dr. Ravichandran and Dr. Sastry stressed upon drug repurposing. So basically a molecule is made for some particular purpose, now whether it can be used for some other purpose. So in this particular case, since the system is ready, you have to just put the molecular structure as an input. Say it's some 10-15 molecules, you just copy paste. So Dr. Sastry also mentioned a linear way of representing molecule in SMILES format. So this is the two-dimensional structure represented in SMILES format. You just copy paste, you first draw the molecule in ChemDraw and copy as a SMILES string, put it into the system. Now you can find out whether this list of compound which is in your collection is a anti-cancer or not. If anti-cancer, for which type of cancer it is more efficient. The next step is you can extract the molecular scaffold from the your input molecules, because you don't know how to find the scaffold. What is scaffold? So you try to generate the molecule in a generic format. Say for example, chlorobenzene, bromobenzene, iodobenzene, etc. So there is a benzene is a common. Then you can say halobenzene. So this halobenzene is a scaffold way of representing the molecule in a generic format. People call this is a Markush representation of molecules, where you put R1, R2, R3, and then in that R1, R2, R3, you will be using what is R1, whether it is the alkyl, aryl, halide. This type of information is mostly represented in patenting and also in some of the publications. They make a generic structure and make a table of table containing what this functional group represents. So we are doing the reverse. Given all molecules as an input, we try to derive the scaffolds and functional groups. So once the scaffolds are built as shown here, now you can scaffold, and this is the building blocks or the functional groups built. Now you can generate the virtual library of huge molecules this combinatorial means. That means all the functional groups here is replaced in particular position R1, R2, R3. So you are able to generate more molecules than your input molecules. Okay. So what is your input molecule? Your input molecule is very few, but whereas your virtual library is huge one. Now, once you have the virtual library, you are able to generate the progressive drug-like score, progressive lead-like score, and then other lead-like values and drug-like scores. Okay. So once you have these scores, then you can prioritize the compound, like how people use Lipinski rule of 5. Similarly, you can use this for shortlisting some of the compounds into the system. And in the beginning of the presentation, earlier presentation also, someone told that the fragment-based drug discovery, they discussed about it's not necessary all the compound fall under drug likeliness. Say, for example, Taxol, which never follows any of these rules. So it is your choice. But the computer gave the calculated and predicted values. It is up to you whether you want to take drug-like compound predicted by the computer, or you want to find the outliers, which you want to do some exceptional studies. You have a choice. Okay. Once you have the list of compound which is in your collection, now you generated the virtual library of novel molecules, and then you have an annotated property whether it's drug-like or lead-like. Once you have a drug-like lead-like, then you can go to cancer type, what type of cancer. I already told you there are about several machine learning techniques and deep learning techniques which are in the which cannot be done with your laptop or desktop. So these mathematical AI techniques are implemented in a remote computer environment where these computers are enabled with several GPU type of cards. Say, for example, in this case, you can select the high performance computing. So nowadays, you don't need to own your GPU computers. You can rent it on Amazon Cloud or Google Cloud, where your job can be running in the cloud. Then you can pay per use. Type of models are now coming up, so you don't need to hold the computer in your resources or in your facility. Okay, so in this case we try to optimize the system such a way that once the inputs like virtual library is generated, the entire virtual library is passed on to high performance computing in the remote location. Okay, once the high performance computing received the data, then it will do the QSIR studies, docking studies, TP score, molecular descriptor calculations, shortlisting, many things it can do. Okay, so you can give the commands here. You should have first to know which computer you are accessing and what input you are sending, what is your credentials. So you have 25 nodes are there. You can select how many nodes you want to do it. Once it is done, now sometimes you don't have the molecules in SMILES format. In our case, we have made the system where even a common name of a drug can be used as an input. So you just copy paste the chemical name or drug name from the drug bank or published literature. So the text is converted into a chemical structure, and then the chemical structure goes through the entire pipeline of identifying scaffold, building a virtual library, calculating the drug-like properties, cancer-like properties. Then once that is shortlisted, those new compounds or known compounds, those can be docked against the target of interest. In this case, it's cancer-specific targets or antiviral specific means viral targets, etc. Okay, so we made the systems right now. Right. So now you can also visualize the molecules. So you just copy paste the molecules. You put the name to structure, and then you can convert 2D into 3D, etc., etc. All things are possible now. Now this system is also integrated with a visualization of docked poses. So you can see the binding interactions, interaction diagrams, how the ligand goes and binds which target. So now you can do the combinatorial docking with respect to this anti-cancer research. So you can select your kinases or whatever the proteins of interest, and you already shortlisted some of the compounds which is known compound or drugs in the context of repurposing or in the virtual library or a new molecules. So once you have the list of known compound and the virtual library, now you are able to do docking studies, and then you can prioritize. So 3 levels I have shown. One, first step is given a molecule, whether it's a drug or non-drug. Second, whether it's anti-cancer or not. If it is anti-cancer, with respect to which cancer, what is the GI50 value, predicted GI50 value? With respect to different mathematical models like machine learning models, deep learning models, which method, which model, what is the accuracy, and then prioritize the molecules. Once the molecules are prioritized, it can be go through this docking studies. It is up to you. You want to dock all the virtual compound against all targets? Yes, you can do it. It depends upon how much resources you have, etc. Okay. So we have a huge resource, so we are able to do this type of calculations in a remote location. And this is just interface which facilitate just by click you are able to do it without much programming knowledge required for this. So finally, you will be getting the what is the active site, what after active site, what is the binding energy of that particular molecule. In this case, I'm just showing, even though we have done for anti-cancer, so here we also tested this against COVID targets, and any target, any ligand, you can do docking in a remote environment, and then you can get the binding energy and process. Okay, so then you can get the histogram of what ranges this binding energy comes. Then you can shortlist the compounds of your interest. Okay, this is able to do text mining from the PubMed literature, identify the chemical names. Once the given abstract copy paste or automatically extracted based on the PMID, it extracts all the molecules with respect to disease of interest. So now most of my presentation is over. I want to tell what actually artificial intelligence, what inspired us to do this work. Okay, how it is applicable into the chemistry. Okay, so you just relax and sit back and listen. Okay, so you know this type of movie people must have seen some 20-30 years back. So this is just movie where in this movie Arnold comes and recognizes the type of bike, and then he knows what model, etc. So my question is, I was so inspired by this. Now in our condition, by drawing a molecule on a piece of paper with pencil, using the web camera, can I recognize this? Okay, can I recognize the molecule? Okay, so visual computing, here the image processing is very important. Here we can say that now Google is using image processing for self-driving car, so etc, etc. Now this was done almost 10 years back, where we tried to do, it was already, now it was sanctioned, US patent also granted. We're looking at the molecule, whether are we able to identify the atoms and bonds. Once you identify the atoms and bonds in 2-dimensional, we were able to do 3-dimensional calculations. Now the next step in AI is just to draw the molecule and show the webcam or your mobile camera or a computer webcam, and in future the computer will be able to tell maybe a drug repurposing context, maybe a anti-cancer agent context. Then you can also say whether this molecule is anti-cancer or not. Just ask this question. Maybe people say Alexa, people say Siri. In future, maybe I may call this as a Chitty, whether this molecule is anti-cancer or not. What will happen? This computer will recognize the molecule which I have drawn based on my medicinal chemistry background and organic chemistry background. Now looking at the molecule, I want my computer should understand exactly like how I understand. Looking at all the available information, 3 million experiment with respect to cancer, any information with respect to COVID, it should tell me whether the molecule which I have drawn on the piece of paper, whether it is anti-cancer. If anti-cancer, what is the predicted bioactivity for this compound? So I want a projector integrated with this web camera, where the projector is showing the bioactivity on the molecule itself. So this is the role of AI. Okay, so this is what we are now working on. Looking at the molecule, first we try to do whether molecule can be recognized. Now the next level will be, it will be projecting back the anti-cancer, antiviral activity on the molecule itself as a projector. Okay, so so now the next step is we also try to integrate these tools in an email platform, because people may not have all the softwares necessary, all the skill sets necessary. So we try to do it in a very simplified form. So if you can select the, okay, I want to do docking studies, you just send a email with one HIV aspirin as the text in the subject line. So you will be surprised to receive an email with all the calculation done with the poses of aspirin in one HIV in active sites. So this is the method. This also we published with patent. So now I want to take you into details. So you must be surprised to see this slide because it is a repeat slide of Dr. Sharada Swain morning he presented. I don't know. I did not take his permission to screenshot, take the screenshot of his presentation. But for the interest of the audience here, I want to show how AI can in future, invasive in future. So you are giving a presentation in a conference like this, where you imagine Alexa is listening, Siri is listening, all people are listening. So they look at the slides, and then since I shown you molecule to activity, I shown bioactivity spectrum prediction for a given compound. Suppose this slide is shown by a presenter across the world, somewhere in the part of the world, in some of the conference rooms. Imagine if the software is integrated in the cloud or system. This PowerPoint goes through that presentation. I am talking about 2030, 2050. I don't know. Maybe because I'm presenting today, maybe next year somebody can come up with the idea. So some PowerPoint presentation showing a molecule on the screen and describing about library of protein kinase inhibitor. He spoke about it, or the computer itself read the library of protein kinase inhibitors, right? What it is supposed to do. Okay, he also talked about this type of presentation. Suppose imagine the computer is able to recognize all these molecular structures in the presentation. Okay, now the interesting part comes. Okay, so sometimes people say that oh, I am listening, Google, I am listening, Google, can you tell me the answer? So in that context, now whatever I am speaking, imagine that Google is listening, Alexa is listening, Siri is listening. Okay, now what happens to the next? That is most dangerous. Okay, then AI is watching. Already we know that AI is watching. It is able to recognize the whether it's a human being, whether it is the object, car, etc., etc. Now what happens to in this particular case? Okay, actually it happened. So this PowerPoint slide containing this kinase inhibitors, protein kinase inhibitors, was gone through several level of interactions. So maybe in the next presentation or 1 year from now, I will be able to show live demonstration of from a video taking molecules and doing some calculations and coming up with properties of those molecules from just passing through the video. Imagine if this is successful in all YouTube video where educational or these chemical structures representations are there. In future, you can extract the information from video also, and then eventually live presentations where molecules can be recognized. Once the molecules are recognized, then it can go through several steps. Here I am trying to show you the molecule is recognized from the PowerPoint presentation, and then it calculates the truly computable chemical structure, which I shown you using the patented technology, which we have done already. Given a molecule as an image, you are able to get the chemical structure. Once you get the chemical structure, you can convert the structure into a chemical name. Once you convert it into a chemical name, which can be a common name, which can be a IUPAC name, etc., it can go and search the PubMed, patents, etc. Then it can also use that molecule 1-dimensional, 2-dimensional, 3-dimensional recognized from the image, from the PowerPoint slides. It can build the library of molecules. Once it built the library of molecule, I already shown you. Once you have a library of molecule, you can find the molecular scaffolds and functional groups. These molecular scaffolds and functional groups can be used for building virtual library and predicting the bioactivity. Then all the calculations will be done in the remote computer where you have access. Maybe your credit card is linked to that. So the next step is very interesting, that the molecules recognize, then convert into SMILES format, convert into 3D structure. This is today's morning presentation slide. this is today's morning presentation slide, which is converted into smiles and then converted into 3D structures. It can go through the pipeline to tell whether the molecules are drug-like or not. If drug-like, whether it's anti-cancer or not. Okay. Then the second, what is very dangerous and risky. What will happen? Because it understood from your slides, these are the compounds which are protein kinase inhibitors. Then it went on its own to identify what are the other protein kinase inhibitors known to the world in the Google or PubMed, all other protein kinase inhibitors it extracted, and then it already knows that what is the source in which the protein kinase inhibitor interactions can be studied. Then it can actually calculate tomorrow. It will have a result which can be uploaded into bioarchives and chemarchive. This is more dangerous. Why? Because the person who did the original research and presented in a conference room did not know that there is an AI working in the back end and doing this type of studies. Okay, don't worry, it is still far from reality. I think you got what is the role of AI in drug discovery, how practically it can be used for designing new molecules from the known molecules, and what is the risk associated with privacy invasion, molecular data being recognized automatically. Actually, molecular structures are $1 million compounds. So if your molecule is escaped, or when you present in a corporate environment, if it can go through this type of systems, maybe who knows, tomorrow your data is already in the hands of Microsoft, Google, and WhatsApp, etc. Okay, so today this is for the benefit of teachers, and because here faculties are there. So today, traditional way, we do the when students are writing exams, we watch and then do the things. What will be the future of AI? The computer will be watching your each and every moment. So you won't feel uncomfortable. Whenever we feel the camera around us, surveillance camera, we feel uncomfortable. Similarly, in future, it will become a new normal, where the students may be monitored and other things. So if you extend this into drug discovery, so what we are trying to do is traditional versus modern drug discovery. Okay, so I am almost completed my presentation. I am at the end of my presentation. All the tools and techniques which I described is already presented in the textbook. This is a unique type. See, I believe in practical rather than just theory. So I want each and every one of you, being a chemist, I can learn some computers and able to do these things. I feel each and every one can learn from these resources, and they can build their own sophisticated tools for a period of time. Okay, so and we also published a book, and we published several set of papers back to back. This role of Chemoinformatics tools in drug discovery. In this case, role of chemical reactivity and transition modeling for virtual screening, and protein ligand complex guided approach for virtual screening. Chemoinformatics for building molecular network. We used marine organisms. And then we also used these machine learning techniques for virtual screening lead molecules. Then pharmacophore based docking, virtual screening. These are all published in journal. And then predictive bioactive compound using NMR. Recently we published a work on identifying molecular metabolites from the medicinal plants. So these are all the tools already published, and what is the mechanism, what is the data, what are the tools. These are all back-to-back publication of all the 10 papers. Okay, some another set of papers are coming shortly, and we are continuously working on it. Okay, so maybe this book also will come into soon. You can see that. Thank you for your patience. And if you have any questions, I can take now.